Okay, so in this second segment, welcome back. In this second segment, we are going to be looking at the different methods employed by scientists, microbiologists to classify organisms. We've seen the classification system that has been put forth. We saw that there were data studies and to be able to identify the differences in organelles, cellular organization, and methods of nutrition. So in this segment now, we are going to appreciate the main methods that I the first is morphology. Morphology. Classification of organisms, especially macroscopic organisms, the number one criteria used is morphology. When botanists go to the field to classify organisms, the first thing is to pick an insect, they look at it, and they start describing the mouth parts, start defining the wing segment, the wings and the abdomen segmentation and stuff. And from there, they're able to classify them. And then they go further to now go to species and the rest. For humans, it is easy for you to see that Jerry is different from James, Charlotte is different from Chanel, and so on. But with microorganisms, bacteria, viruses, and so on, the first thing is that we need a microscope to be able to study them. We cannot just look at one and say it's different from two. It doesn't work like that with them. So the first thing we want to still use is morphology. Of course, shape is very important in identifying anything. So when we look at the basic shape, the size, characteristic arrangement of organelles, we can already start having an idea what this organism could be. The presence or absence of a flagella, the presence or absence of a capsule, the presence or absence of an endospore also goes a long way to help us differentiate these organisms. Let's take, for instance, the typical method used in classifying bacteria. The typical method used in classifying bacteria based on shape. So let's say we're looking at the bacteria. So we look if the flagella has a flagella. Flagella is an organelle used for locomotion. So if you have a flagella, it means that what? You are motile. And if you don't have a flagella, you are non-motile. So we can either observe a flagella or what? We use motility to classify the organisms. Capsules are organelles that are used to evade the immune system. They are very, very important in opsonization. So the presence of a capsule will mean that the organism could be more what? Pathogenic, while the absence of a capsule means that the organism will easily engulfed by opsonization. So capsules evade opsonization. We can also look at spores. By the way, the presence of flagella is one thing, and the positioning of the flagella is also employed. Is it a single flagella? Are there two flagella located at opposite poles? Are there flagella surrounding the entire cell? Those are all methods that are employed. Spores, the positioning of the spores, is it an endospore? And if it's an endospore, is it a terminal spore? Is it a mid-spore? And so on. So these are all morphological aspects that are brought together. But more broadly is the shape of the bacteria. So it can either be oval in shape. We call it a cocci. And if it is a cocci, the shape can be, the organism could exist in pairs. An example is what? An example is Neisseria gonorrhoeae. This is a diplococci. They could exist in chains, and this is what called a strep. An example is Streptococcus leukocytogenes. They could exist in bunches, and this case we call a staph. An example is Staphylococcus. I've already mentioned aureus before. This is it about cocci. The bacteria could also exist in rods, right? So the rods are broadly classified as either a gram-positive or a gram-negative rod. Gram-positive rods are further divided as either sporing or non-sporing. To be further divided as aerobic or non-aerobic. Okay, so we're saying that it's a gram-positive aerobic spore former. In this case, we'll be looking at what? The genus Bacilli, Bacillus anthracis. Bacillus anthracis is an example, which causes the disease anthrax. It is gram-positive, non-aerobic spore forming. We could look at the Clostridia. An example is Clostridium tetani, the causative agent of tetanus. It could be gram-positive or non-sporing. They will look we could look at Corynebacterium diphtheria. Corynebacterium diphtheria that causes diphtheria. So we could have gram-negatives. Gram-negatives could be a member of the Enterobacteriaceae. So we're looking at Salmonella, the causative agent of typhoid, Shigella, the causative agent of shigellosis, and so on. Or they could be non-members of the Enterobacteriaceae. We're looking now at Campylobacter, and so on. While we could also have the comma-shaped bacteria, which we call Vibrio. And the typical example is what? The Vibrio cholerae, that causes cholera. It could be spiral in shape, called spirochates. An example is what? Treponema purulum, that causes what? Syphilis. So these are different ways by which bacteria are classified based on shape. These are the different ways in which bacteria are classified based on shape. And just on this shape, we can be able to identify which bacteria we're dealing with. No, presumptively identify. Just having it as a gram-positive diplococci does not immediately tell us that we are dealing with Neisseria gonorrhoeae. There are many other characterizations that we need. Just having it as a gram-negative rods belonging to the Enterobacteriaceae group, we need to further differentiate it, whether it's a Salmonella, Shigella, Proteus, and so on. Okay, so this is just the first basis for the classification procedure. The next thing is differential staining. Differential staining takes advantage of the fact that the different microbial source organelles absorb stains differently. Organelles of the microbial cell absorb stains differently. And therefore, if we explore this differential absorption of stains, we can be able to visualize the organisms differently. The main and broadest staining technique that is employed in bacteriology is what we call the gram staining. In other courses, and in this course, you are going to go deeply into the gram staining procedure. But just to highlight that gram staining broadly differentiates bacteria as gram positives due to the presence of a thick peptidoglycan layer, or gram negatives due to the presence of a thin peptidoglycan layer. So you must have heard me already when I was classifying based on shape to further gram positive or gram negative. So when you are able to, first of all, broadly classify organisms like gram positive, gram negative, enables you to go further with other analysis. The next staining technique that could be very important in bacteriology is the acid-fast stain. Acid-fast stain helps us identify the presence of a mycolic acid layer, which is typically present in Mycobacterium. So Mycobacterium tuberculosis, Mycobacterium leprae, and other species have this layer that resists decolorization even after acid-fast stain. So with this staining, we can go to already visualize whether they are gram positive or negative, whether they have an acid-fast stain. And staining also helps us demonstrate the presence of a capsule, the presence of spores. Okay, so these are all techniques that you'll be exploring when you're doing staining. So have that in mind. Biochemical testing are also employed in the identification of bacteria. The rationale here is that when an organism is able to a nutrient, it is because it is able to synthesize a particular enzyme, and also because it possesses genes that allows it to synthesize those enzymes that utilize those nutrients. So by exploring, maybe in growth medium, the ability of a microorganism to utilize a particular nutrient or substrate, it helps us already presumptively say this organism has these genes, or this organism produces this enzyme. Okay, there are so many different growth presentations that we can identify just by observing the ability or inability of organisms to utilize certain substrates, and also the ability to release certain byproducts. Okay, it is exploited in what we classify as biochemical testing, and it's very important since it is another step further in identifying for classifying organisms. Serology is also employed. Serology here will refer to the fact that, or we are exploring the fact that when an organism enters the human body or animal body, the first reaction is that it releases an antigen, and this antigen provokes the human body to react by releasing an antibody. Because antibody reaction and production and certain reaction is very specific, we can identify specific antibodies produced to further identify the antigens that provoked its production. Or we could identify antigens and move towards antibody. Okay, either ways. So we are exploring the fact that this reaction is very specific. And since it's very specific, we can use it in identification and subsequent classification. This can be done by observing for agglutination, precipitation, hemolysis, and so on. So there are different ways with which we can observe this. We'll be doing some of these tests, I think, in this course as well. Phage typing. Phage typing is a technique that is gaining ground and has become one of the principal methods used in identifying organisms. This phage typing technique takes advantage of the fact that certain viruses infect particular bacteria or particular species of bacteria. A phage is a virus that infects a bacteria. A phage is a virus that infects a bacteria. And it has been observed that phages are very specific in the bacterial either family or species or genus or genera that they infect. They are very specific with this. So we can demonstrate viral infectivity in different bacteria and therefore use it to identify and classify the bacteria. Let me take for instance, there are certain viruses that infect all members of the coliform. Coliform are bacteria that live in the gastrointestinal tract. So viruses have been shown to specifically infect this line of bacteria or this group of bacteria, and therefore are grouped as coliphage. So if you identify a coliphage and use it to observe its infectivity in a particular bacteria, we can confirm that that bacteria is what? A coliform. Okay, this is just an example, but this is how the procedure is explored. DNA-based composition, I'm sure you must have heard in cell microbiology, in cell biology, that the GC content of organisms is very, very constant and could be explored in identifying organisms. So if we analyze the GC content of different organisms and observe similarity in the percentage of guanine to cytosine, we could presumptively say that those organisms are similar or are related, and we could further do other analysis to confirm this relationship. But GC content is very, very important, and it is constant across species. DNA fingerprinting is another method that is used. In DNA fingerprinting, we are exploiting the fact that restriction enzymes cut the DNA molecule at very restricted areas. Restriction enzymes cut DNA molecules at very restricted areas. We know that these restricted areas are also found in different species. An equal R1 cutting region. So let me take an example. If this is a DNA molecule from one bacteria, and a DNA molecule, from another bacteria, we could use the same cut. We could use the same restriction enzyme to cut these two molecules. So if it cuts here and cuts here, in this same organism, it cuts here and cuts here. And for the third organism, remember we are using the same restriction enzyme. It cuts here and here. If we run electrophoresis on these three DNA molecules, we are going to see that this strand, this area, is going to be similar only in two organisms. This one might be similar in the three, but this one will be similar only in two. This length, this one I'm referring to as this area, this length will be similar only in two organisms. So what does it mean? It means that these two are more related than they are to this third. Okay, so the restriction fragments generated from these two are identical, while here only a proportion of the restriction fragments are identical. So we could use this to explore relationship between organisms and classify them as such. Sequencing techniques have been explored now, are currently being explored. This is the latest advances in molecular biology. We can sequence the entire organism now and identify the similarity between the bases and also elaborate it to mean relationships or lack of relationships. So this is the new world of science now. Sequencing, next generation sequencing is the mainstay now. Polymerase chain reaction, PCR. All of you are now familiar with the word PCR thanks to COVID-19, that you need to do a PCR test. So PCR helps identify a sequence in an organism. I'm sure you guys must have done PCR, but let me just throw some light on it in case you are new. So we're saying that these are two sequences. Sorry, this is a DNA sequence. The first assumption is that this is a double-stranded molecule. Okay, this is a double-stranded molecule. And under ideal condition, it is bound to each other by what? Hydrogen bases. Each strand is bound to each other by hydrogen bases. So conditions can cause this binding to break down. One is the presence of the enzyme helicase. If helicase is present, helicase will break down these bonds and open it during DNA replication. Another reason why this will break down is heating. If we heat this up, hydrogen bonds are very weak bonds. They're going to break down, and we can also explore it. So when these conditions that caused the hydrogen bonds to break down cease to exist, when these conditions are removed, the DNA molecule is also going to come back and hybridize, anneal. It's going to anneal, and then the double-stranded molecule is going to regain itself. So what are we now doing here? In PCR, the first thing we do is that we separate DNA molecules from different organisms. Okay? When we separate DNA molecule, we try to allow them to bind. We try to allow them to bind. First, by providing a primer that is specific to the organism we are interested in. Binding of two strands and synthesis of a new strand greatly requires a primer. I think more details on this will be done in other lectures. But I'm just trying to throw light. I don't have enough time to explain this detailing. So I'm saying that the synthesis of a new strand of DNA after the typical unwinding by helicase requires a primer, because DNA polymerase 3, which is the DNA synthesizing enzymes in prokaryotes, does not have the ability to initiate polymerization. It cannot start from 0 nucleotide. It needs to only add nucleotide to an existing N. So if this N is present, then synthesis will occur. If this N is present, synthesis will occur. In a typical scenario, when we provide the primer, so for instance, we want to diagnose hepatitis B. So I've taken samples from the patient's serum that was supposed to contain different organisms, including hepatitis B. But we separate all these DNA molecules by heating. We break down the bond by heating. After we do that, we now provide only a primer specific for hepatitis B. We'll provide only a primer specific for hepatitis B. And if a hepatitis B sequence or strand was present, if a DNA strand, sorry, I'm using hypothetical because it might not necessarily be the case. If a strand was present for hepatitis B here, the primer that was specific for hepatitis B will bind here, and therefore prolongation and duplication will occur. But if this hepatitis B strand is not present, this primer will not bind. And this primer was specific only for this hepatitis B. So despite the different DNA molecules and strands that are present in our mixture, this primer will not be able to bind to any of them. And because there's no binding, there's no synthesis of a new strand, and therefore we cannot observe duplication in PCR mixture reactions. I'll be able to explain more on this if you have queries in the course of it. The next procedure is used is nucleotide hybridization. Hybridization also takes that. Okay, so when we separate the DNA molecules, they can only anneal themselves if they are related. So in hybridization technique here, we could bring from two different organisms and observed if they will anneal. And as indicated, annealing means there is relationship. different things are explored in this. We have Southern blotting, which is a very popular technique used in identifying and classifying organisms. So unfortunately, these are very broad procedures that I can't explain within minutes, but I'm hoping that you get the basics of it, you get a basic representation of it, and from there, we can be able to move further. I want to start with I want to say that we could start with questioning now, and then the lecture will stop